Hi everyone, Dr. Hall here. And so 37 Part B, we're going to talk about gametogenesis. Now that we've done an introductory lesson in basic genetics, let's talk about how we actually make gametes. So let's remind ourselves that chromosomes in our cells, there's 23 different types, and we have 46 total because we have a pair of each. And you'll recall that one copy came from the sperm and the other one came from the oocyte, right? So for chromosome 1, I have 2 of them. One of them came from my dad's sperm and the other one came from my mom's oocyte. So when we're making gametes, when we're making sex cells, when we're making sperm or oocyte, also called ovum, then each of those cells needs to have just one copy of each chromosome, which we call haploid. So what we're going to talk about today is how we actually do that. So gametogenesis, if you kind of take a look at the word, you go, oh, genesis, that's creation, right? And gameto, that means gametes. So gametogenesis sounds like a really fancy word, but it just means creation of gametes. So when we think about cellular replication or producing cells to begin with, there's no such thing as spontaneous generation. You can't just pop a cell out of nothing. All cells have to come from other cells. So making new cells, therefore, is called replication because you're making a copy of an already existing cell. Most of the time in our bodies, when we're growing, when we're healing, when we're doing repair work, maintenance work, replacing cells as they die off normally, our cells are making exact copies of themselves, and that process is called mitosis. So you take one cell and you end up then basically making a duplicate copy of it, and you end up with 2 what are called daughter cells. So one cell becomes 2 identical daughter cells. For making gametes, however, we don't want an identical copy. We have 2 goals, actually. So the first one we've talked about, which is that we need to have the genetic material. We need to only have a half C cell, a haploid cell with 23 chromosomes, because it's going to meet up with another gamete to then form a new set of genetic combinations that's never existed before when we fertilize the egg. The other thing that we're going to want to do with meiosis is we're going to mix up the genes a little bit from one chromosome to another so that there are different combinations of genes on the same chromosome from what we had before. So meiosis is the creation, therefore, of some diverse cells that are haploid, that only have half the usual number of chromosomes, or 23 in the case of humans. For our purposes, so there will be other classes where you learn meiosis 1 and meiosis 2, and you learn about prophase and metaphase and anaphase and telophase. We're not going to go into that level of depth because I want to focus on concepts. So for our purposes, the 4 steps involved in meiosis are as follows: duplication, crossover, first division, and second division. And we're going to go through those one by one. So first, I want to introduce you to our starting cell. So for the purposes of this demonstration, which alas, we cannot do in person, or else you would all have popsicle sticks just like this. But for the purpose of this illustration, this is a fictional cell that instead of having 23 pairs of chromosomes, it only has 2 pairs of chromosomes. So it has the big chromosomes and the little chromosomes. So this organism has only 4 chromosomes total arranged in 2 pairs for simplicity. And then you see these little things that are stuck onto these chromosomes. So we have a gene for star color, a gene for hair color, and a gene for smiley face color. And you can see that on different chromosomes, they can sometimes be the same, right? These both have a gene for blonde hair, but this one has, blue star color, and that one has pink. This one has a pink smiley face, and that one has yellow. If we look at our small chromosomes, right, so we've got eye color. These are supposed to be little beady eyes. We've got eye color here. It looks like they're both green or blue, kind of hard to tell. And then we have heart color here. You see the little heart stickers. The one on the left is blue, and the one on the right is gold. So this is our existing starting point. This is our diploid cell. This is what we're going to begin the process of meiosis with. So our first step is duplication. Step 1 is duplication. we're going to make exact copies of all of the chromosomes that we have. So you can see here, I've made an exact copy of the big pink, I've made an exact copy of the big blue, exact copy of the little pink, and an exact copy of the little blue. This may seem really counterintuitive. The whole point was to decrease the number of chromosomes, and here we are doubling them. I know, it seems ridiculous, but yet this is how it happens. So step 1 is duplication. Step 2 is called crossover. So what happens is they're called homologous chromosomes. So the chromosomes that originally were the ones that were kind of inherited from the oocyte, for example, are about to have to say goodbye to the chromosomes that originally came from the sperm. So what they're going to do is they're going to cross over. They're going to lay kind of on top of their homologous chromosomes, the big ones on top of the big ones and the little ones on top of the little ones. And when they do this, they're going to exchange some of their genes. So some segments of their DNA are going to get exchanged between one and the other. It's almost like, I know we're going to have to say goodbye to each other, so here's a little memento of me. Here, take this token so you'll always remember me. So at the end of crossover, you can see, look, this pink chromosome ended up with a gene from a blue one. This one ended up with a gene from a blue one, right? So we just mixed things up a little bit, and same thing with the small chromosomes over there. So what we've done is we've changed things up so that the combination of genes on any one chromosome is actually already different than the combination on any other chromosome, which is pretty cool. So crossover is where we introduce that genetic variety. Step 3, then, we are going to split these guys up. So I told you it was coming. So first division, all the pinks are going to go to one cell and all the blues are going to go to another cell. So if you originally were one of the chromosomes inherited from the oocyte, you're going to come over here. And if you originally descended from the sperm's chromosomes, you're going to move over here. So now we have 2 separate cells, but each still has 4 chromosomes arranged in 2 pairs. So we've accomplished the one objective of increasing genetic diversity, but we haven't yet halved the genetic material. So now we go to step 4. Step 4 is the second division. So now each of these cells is going to split into 2 cells, and each of them gets 1 big and 1 little chromosome, right? So at the end of meiosis, we started with 1 progenitor cell, and we ended up with 4 half-Z cells with increased genetic variation in terms of their combinations. So here we go. These are our gametes. They are half-Z cells with the genes all mixed up a little bit because variety is the spice of life, right? We are sexually reproducing organisms. We keep trying different combinations to see if maybe we're going to hit on a combination that's especially fit for survival. So as you can see, if you look at these, no one of these cells has the exact same types of genes as the other, right? Some of them have yellow smiley faces with gold hearts, but one has a yellow smiley face with a blue heart, right? And of the pink smiley faces, one has a gold heart, another one has a blue heart. So everything is mixed up. No one of these cells has exactly the same combination of genes. That is the goal of meiosis. So just a reminder, all cells come from other cells. Mitosis is what you might remember from high school biology, and that is what we use for normal growth, upkeep, maintenance, and repair. Meiosis we use only for the creation of gametes, our sperm, and our oocytes. And we end up with 4 cells, each of which is haploid, or N, which means it has 23 chromosomes. So in the case of sperm, right, the same thing is going to happen. We start with what's called a spermatogonium. It undergoes those steps of meiosis, and we end up with 4 sperm, each different from each other, and each of which is haploid. Here's another illustration of that same process, and I've kind of put in the stages that I'm asking you to know. So you can kind of see there's that crossover. They're exchanging some genetic material. Now you can see how they've swapped genes from one to the other, right? So after we end up with those 4 cells from spermatogenesis, those cells are then going to change shape. And they're going to slim down. They're going to get rid of any extra organelles because they're really just focused on becoming lean, mean swimming machines that can deliver those chromosomes. And so any cellular organelles that don't help them do that, they're going to get rid of. So they get rid of their Golgi apparatus. They get rid of their endoplasmic reticulum. They're getting rid of all that stuff and getting rid of a ton of cytoplasm so they can form this aerodynamic shape. So there's a head of the sperm, which will contain the nucleus, and which is where the chromosomes are, of course, and then an acrosome, which is like a little cap sitting on the top of the sperm there, which contains enzymes, which are going to be important for penetrating and getting to the inside of the oocyte. We then have this mid piece, which is chock full of mitochondria. So that's the one organelle that the sperm are not going to get rid of because they're going to need a lot of energy. They're going to need a lot of ATP because they are going to do a lot of swimming. They're going to swim hard and fast and far. So their mitochondria are going to produce ATP in order to power their tail, which is a flagellum. So remember when we learned about the different parts of cells in the beginning of the semester, it's a flagellum made of microtubules, you might remember those, that allow it to swim. And so you'll also recall that in semen, we learned that the fluid secreted by the seminal vesicles and the prostate to form semen contains fructose. And so fructose is what those mitochondria are going to use to break down through aerobic cellular respiration and form ATP, produce ATP. Now, what about for oogenesis? So oogenesis, or the creation of an oocyte, is going to go through the same 4 steps as spermatogenesis. However, we're only going to end up with one finished mature oocyte. What happens is when we do those divisions, those 2 divisions, one of the cells gets no cytoplasm and no organelles, and it's called a polar body, and it will just die and disintegrate, right? So that's for the first division, and then when we do the second division, again, right, it just becomes something called a polar body because that oocyte needs to hold on to all the organelles to survive. Because remember, the sperm isn't bringing anything, right? He's not bringing the bath towels or the toaster oven or the frying pan or the shower curtain. He's not bringing any of the stuff that you need to run a household, right? He's just getting the chromosomes there. That's it. So the oocyte has to have all the things that you need for housekeeping for a cell. The other reason that we only want to have one oocyte, as I mentioned when we did the female reproductive system, is that humans are not built for multiple gestations. So we don't have room in our pelvises, which are relatively narrow because we walk upright on our legs. Our pelvises are relatively narrow. We don't have enough room for lots of babies in there, right? So even people with twins, it's very rare for them to make it all the way till their due date because there just isn't enough room. And so those babies are almost always born prematurely, sometimes very much so, usually just mildly so. So we only want one oocyte, so that's a key difference. Then between spermatogenesis and oogenesis is that spermatogenesis, you're going to form 4 gametes for each of these processes that you do, but for oogenesis, we're only going to make one oocyte. And so from here, we are going to then move next into what happens when sperm meets egg, otherwise known as fertilization, and that'll be part C of the lecture.